Hi everyone. So in the prior videos, we talked about how NK cells and cytotoxic T cells can interact with our cancer cells. We're going to move on to talking about how cancer can avoid the immune system. And it's going to have a few different tactics of how it's able to do this. We'll just jump right in to the first option. So in order for cancer to be detected by a cytotoxic T cell, I just draw out my tumor cell really quick. Part of the interaction that occurs is having this MHC1 molecule present an antigen, which is recognized by the T cell receptor. And this is our MHC1. So this interaction is essential for our CD8 plus to essentially recognize our tumor cell. So one thing that target or tumor cells can do is get rid of this antigen. I'm just going to draw an X over this. So we can remove antigens from the cell surface. So when we get rid of those antigens, our CD8 plus cells can't recognize this tumor anymore. They don't have anything to bind to and recognize, so we cannot attack. That's going to be our first way that we're able to avoid kind of detection by the immune system. All right. Second method. If we grab our CD8-1 and we grab our tumor cell again, okay. So in this situation, we're actually just going to draw out kind of the same schematic. We have our MHC1 presenting an antigen, which is recognized by our tumor cell receptor, or our T cell receptor, sorry, not tumor, T cell receptor. In this case, instead of getting rid of the antigen, we're going to get rid of the MHC1 molecule. And we are going to keep our MHC1 molecule inside the cell instead of trafficking it to the cell surface. So this trafficking process is really what's blocked in this case. So I'll just describe that right here. So we don't move MHC1 to the cell surface. So again, our CD8 plus can't recognize an antigen without having an MHC1 molecule to present it. So our CD8 plus cell isn't going to be able to recognize our tumor very well in this case. However, an interesting side effect of this, if I draw and kind of go back to one of our other cell types that we have to worry about, that we talked about, which is our NK cell. If we don't have any MHC1 molecules on the cell surface, our NK cell is going to think that that is a foreign cell and it's going to attack. So this is going to protect us from our CD8 plus cell type, but it's not going to protect us from our NK cells. Okay. Moving on to our third tactic. Again, if I kind of copy these cells down, go. All right. So in this case, actually, I'll put an NK cell here instead because this is going to be more relevant for NK versus our CD8 plus. So we talked about previously how tumor cells can have these stress ligands on their cell surface. The main one is called mica that we're going to be talking about. Again, these are stress ligands. And when NK cells see a stress ligand on the cell surface, that lets them know to attack. What we're going to do to get around this is a couple different things. We are going to, first off, decrease the presence of mica at the cell surface. So we won't have as much on our membrane for NK cells to recognize. And the second thing we're going to do is secrete mica into the extracellular fluid. And these are going to kind of act as a decoy, where if we have our NK cell and we have all of this kind of soluble mica surrounding it, our NK cell isn't necessarily going to know that this mica isn't attached to a cell. So it's still going to be binding and recognizing these mica molecules. And because its receptors for this molecule, I'll just draw kind of some receptors for this, are going to be saturated. If we have, for example, a cancer cell up here, it's not going to have any available receptors to recognize that cancer cell. So it's essentially distracting our NK cells and tiring them out with these decoy stress ligands. So that's going to be kind of our third way that we're avoiding the immune system. And if I scroll back up, there's kind of one other main action that we're going to take. We are going to, if I write out my kind of fourth bullet point here, we are going to decrease the presence of our FAS receptor. So we talked about earlier how this is used to signal extrinsic apoptosis. So now when we have a CD8 plus that has this FAS ligand on it, when it tries to bind to a tumor cell, it's not going to have anything to bind to. So it cannot cause extrinsic apoptosis, cannot signal apoptosis extrinsically. And in conjunction of this, we're also going to increase production of IAPs. IAPs are going to be inhibiting apoptosis. So we're going to increase inhibition of apoptosis via our increase of these IAPs. And then lastly, we do have some fast receptors on most of the cells in our body, and that includes our CD8 plus cells. So we have fast ligands on the surface as well as fast receptors. Something that the tumor is going to do is it's going to secrete fast ligand that can bind to this fast receptor and induce apoptosis in the CD8 plus cell. Along with that, we're going to secrete a couple other molecules. We'll secrete interleukin 10, I abbreviated IL-10, and we're also going to secrete TGF beta. Now, these two molecules are going to be kind of increasing the immune activity, and they're going to be activating Tregs, which, as their name kind of implies, are these regulatory cells that keep our immune system in check. So this is kind of cancer's offensive way of fighting back and saying, not only am I going to remove a lot of my own receptors and molecules, I'm going to start to secrete some molecules that can actually hurt the immune system as well. All right, that was my overview of kind of how cancer can fight back and avoid the immune system.